Okay, so how cells reproduce. So, we're going to talk about two things here, mitosis and meiosis. Or for here, I'm going to pronounce it meiosis, but that's the two things we're going to talk about. We're going to first start with mitosis, though. Okay, so the idea here is this, that our cells can grow two ways. Okay, let me say this. Okay. How cells reproduce. Okay, so we're going to talk about the process of mitosis. Now, for us to become larger organisms, we have two choices. One, our cells can become bigger, or two, we can make more cells. We talked about this before in the first part of the semester, where we are limited in the size that cells can become because what? That surface to area ratio concept. Okay, so there's a limit about how large a cell can become. So that leaves the other part. If we want to grow, we've got to make more cells. We've got to make exact duplicates. And that's what mitosis does. And we're going to talk about the steps involved in mitosis. Now, there's two things. There's mitosis and meiosis. Now, I'm going to pronounce meiosis as meiosis to differentiate the two. But if you take biology or upper-level biologies, be prepared to know that mitosis and meiosis are the two steps, not meiosis. Depends on who you have. But meiosis and meiosis are interchangeable. Okay, but for now we're going to talk about mitosis on this part, and then meiosis or meiosis later on. So these are the two ways that cells can divide. Mitosis, the subject of this coming discussion, is going to focus on making two exact copies of the cell. And mitosis specifically deals with how we separate the nucleus to make sure that each new cell that we create has a perfect complement in the nucleus. Meiosis deals with how we divide up our DNA and our gametes and ultimately produce gametic tissue. Now that also deals with how we introduce variability, and we will talk about that soon. So why mitosis? Well, gosh, we've got to get bigger, taller. We have to replace dead cells because we are in a constant state of death. Our cells are constantly dying, such as in our skin. Okay, so we've got to become larger, and we have got to replace dying tissue. And that's why we do mitosis. To become bigger and to replace the tissue, the cells that are in a constant state of death. Okay, so before we can talk about this, we've got to talk about chromosome structure, how our chromosomes are arranged. We have 46 separate chromosomes. Okay, we have 46. A human would have 46, barring some type of error, mutation, or missing chromosome. That does happen. So, for example, trisomy 21, 3 of the 21st chromosome. So we have 46 chromosomes, typical human complement. But, you see right here, we have, what, 23 chromosomes shown. Why the difference? Well, the difference comes because these are 23 chromosome pairs. And each pair we refer to as homologous chromosomes. So let's look at number 1. We see 2 chromosomes there. One comes from your father, one comes from your mother. Second chromosome, same story. One from your father, one from your mother. Okay? With your chromosomes, you will have 46 chromosomes typically. 23 of those comes from your mother, 23 of those comes from your father. Now we refer to this as a diploid complement, or something that is diploid, di. Di, what does that think of? 2. Okay? In your diploid cell, you have 2 of each number of chromosome. Okay, so you have 2 of the first chromosome, 2 of the second chromosome, 2 of the third chromosome, all the way to the 23rd chromosome, the XY pair, or the XX in females. Okay, Now that's what we refer to as diploid. You have two of each type of chromosome, one from your mother and one from your father. If a cell was only to have one of each chromosome, sperm or an egg, we would call that haploid. We'll talk about that with meiosis. Now for connecting this to mitosis, before mitosis, a cell, for example a human cell, would have 46 separate chromosomes or 23 paired chromosomes. After mitosis, we will have two cells, each with, again, 46 separate chromosomes, or, said another way, 23 paired chromosomes. So let's talk about chromosome before separation. This is a chromosome before duplication. Now, if we were to look at this on a molecular level, it would be like wound up upon itself. It's not one helix of DNA, it's wrapped upon itself. But we're going to use this to represent a lot of the DNA. So before mitosis, we have one chromosome. What happens, though, is before we have the separation, we have got the chromosome duplicating itself. Okay, this is an exact chromosome duplicate. So we have two, it's like party balloons, right? If we compared the sequence in one chromatid and the sister chromatid below it, they would be identical, barring any type of mutation. What's going to happen is, going from 1 and 2, this is in one cell, right? And what's the idea? We're trying to go from one cell to two cells. Because we're going from one cell to two cells, we've got to make a perfect copy of the DNA, such that one of the cells gets one copy, the other cell gets the other copy. And that's the point here, that before mitosis occurs, we have got to duplicate the DNA such that we can separate it into two cells. Okay. Now, these are connected at the centromere, and later on we'll show where microtubules connect here to another structure. But the point is this, I want to make this point. Eventually, toward the end of mitosis, this structure will separate. Now this is the chromosome structure. Okay. Point here is that DNA is in different stages based on the need of the cell. Sometimes it's in the form of chromatin, which is kind of like spaghetti where it's all unwound. You can see the DNA. Other times it's condensed. Condensed is better for situations where you need to move the chromosome. For example, mitosis, that's shown on the right, and the chromatin on the left is good when you can actually need to see the DNA. Okay, need to interact with the DNA on a molecular level. So again, one is good to using the DNA to make protein. The other one is good for when you need to move it around. Now, at the start of mitosis and meiosis, we're going to see the DNA in the cell condense. Okay, we will see it condense. Now, if we were to take, remember we showed you that it was like a party balloon before, and I said that's not a great picture of it? That's because that really represents the DNA that's wound around itself. Okay, DNA in the chromosome is made up of superhelices upon helices. Okay, so what does that mean? That means if we were to take the DNA, right, and wrap that DNA around histones, and those histones, like beads on a string, they wrap around themselves, and that further wraps around, and that further wraps around, and it has these levels of compaction. Okay, the DNA wraps and wraps and wraps again. And this wrapping allows us to compact our DNA into very small sizes. For example, humans have about, if you took all of the DNA in a human and stretched it end to end, you would have about 10 million miles of DNA in all of our cells. Okay? So how do we compact 10 million miles of length down to something that is, very small, such as our human body, right? Well, that's where this compaction comes in, where we wrap our DNA and wrap it further and further and further until we compact it to a very small size. And that's the point, is how do we compact our DNA? We form these super helices and we compact our DNA by wrapping it at multiple levels. We'll talk about that later. Okay. But after, we have to separate the chromosome. Okay, we have to separate this. Remember we said that before this we have one chromosome. It makes a perfect copy, makes two sister chromatids shown in the center. And then eventually those two sister chromatids separate, making two perfect copies of the chromosome. We have to do this in mitosis. How we do that is if we have a point of attachment where we put microtubules and we're going to physically wrench and physically rip apart those two sister chromatids, such that as the cell separates, one side of the cell gets one of those chromosomes, the other side gets the other chromosome. I'm sorry, I had to correct. I said we had 10 million miles. I'm sorry, I was thinking 10 trillion cells. We have 1.5 million miles of DNA. My apologies, that's obviously not on the test, but this kind of gives you an idea of the length of the DNA in a person. Okay? We have about 1.5 million miles of DNA in us. And the reason we can do that, okay, is this idea of supercoiling, where we coil our DNA, and that coil then coils upon itself and coils upon itself. Great picture of that is right here. Okay. We can compact our DNA by coiling it upon itself. And this allows us to really compact and shrink down our DNA to a manageable size that we can fit it in the cell. So chromosome number is all of your total chromosomes. Okay, we have 46 chromosomes, 23 from mom, 23 from dad. Okay, and that's that idea of being diploid. We talked about that before, right? If you have 46 chromosomes, okay, 23 from your father, 23 from your mother, that cell is diploid. Di 2, right? 2 sets. 2 copies. Now, mitosis is not the only issue here, right? Mitosis, we're thinking about how the nucleus separates. But there is a sequence of events before mitosis occurs, and that is interphase. You should know interphase. Interphase is the actions the cell takes in order to duplicate itself before entering into mitosis. Okay, so interphase G1, that's when all the cellular components duplicate, except the chromosomes / DNA. S, synthesis, that's when the DNA replicates. And G2 is the final preparatory steps. And all of these fall under the cycle of interphase. Interphase is when, and let's think about what's happening here. Let's step back and say what's happening. We are going from one cell to 2 cells, right? That's the main idea. We're moving from 1 cell to 2 cells. If I'm going to replicate 1 cell and make 2 cells, guess what I have to do? I'm going to have to first replicate all the internal parts, the mitochondria, the rough endoplasmic reticulum, the Golgi body. All of those things have to increase in amount because I'm going to separate those into two cells. So we've got to make more of those. That's part of interphase. I'm going to separate the genome, right? I've got millions of base pairs of DNA. What can I do with all those millions of pairs? Well, I gotta have a perfect copy of that genome, the DNA in it, because I'm going to separate a cell and it has to go to each one. That's where S comes in, synthesis. We're going to copy and make a perfect duplicate of the genome. G2 is the final steps, but all of these is referred to as interphase. This is the cell duplication steps, both the internal structures and the genome. Now, cancer can come into play here because cancer is the uncontrolled cellular replication process. There's no checks and balances. Many times, a cell will stop at interphase and not continue making more cells. But when these checkpoints fail, cells will continue through interphase, multiply, divide, and that can be the core of what causes cancer. So let's step back here and say, interphase, the cell is doing its internal divisions. It's getting ready to divide. Now, mitosis focuses on the division of the nucleus, which happens right before the physical separation of one cell to two. So we've talked about how the cell replicates its internal components. Now we're going to start dividing the nucleus. That is mitosis. Here's a good summary slide. The first step is prophase. Prophase, the chromosomes condense. We talked about that a few minutes ago. We've got to physically move around our chromosomes. How do we do that? Well, the answer to that is we pack them and tighten them up real close, right, so we can move them around. You'll see why that's important in a minute. 2, the nuclear envelope disappears. Why is that important? Well, we've got to grab on to those chromosomes. If they're in a protective shell, the nuclear envelope, we can't grab and connect onto them. We cannot move them around. So we've got to get rid of that surrounding shell, the nuclear envelope, so we can grapple on to the DNA. Okay. So that's prophase. Metaphase. Look what's happened. Let's compare prophase and metaphase. Do we see the chromosomes are condensed? Yes, we can move them around. Do we see how that nuclear envelope has disintegrated? Yes, we can now grapple on to the DNA, moving it around, ultimately splitting it apart. Think of attaching chains onto something to rip it apart. That's a good analogy. Metaphase. Look at what's happened. See those red lines? Those are microtubules. We have connected microtubules to our DNA. Why is that important? Well, first let's say why we can do that. We can only do that because what? We caused the nuclear envelope to disappear. We got rid of that nuclear envelope. Now we can see the DNA. We can grapple onto it. Now we're moving it around. Why can we move it around? Because we condensed the DNA and now it's in a mobile, movable form. Okay? So metaphase, we're going to take the DNA and line it up. We call this a metaphasic plate. And we could only do this because the DNA is condensed and the nuclear envelope went away. Last part. Okay. So now what do we want to do? We've lined our DNA up, and now we're going to split it. We go from one cell, and now we're going to split the DNA such that each side of the cell gets some DNA on each end. And that's anaphase. Okay. Next step is telophase. Okay? What happens? Well, our chromosomes begin to decondense. Why? Because we don't need to move them around. We're done moving them around. The nuclear envelope reforms because, we don't need to see them with microtubules anymore. We're done with that. Okay, so we can now form that protective shell around them. And those are the 4 steps to mitosis. Know all of the steps in order. Finally, we separate the cell through a process called cytokinesis. Okay, if you go back to interphase, here, see the cytokinesis is the last step and it makes 1 cell into 2. That's the final separation of cells to make 2 cells. Okay, so going back to this image, let's review. G1, interphase, cellular components duplicate. S of interphase, synthesis, DNA replicates. G2, final preparatory steps before mitosis. That is interphase. Interphase is followed by mitosis. Mitosis is concerns with the division of the nucleus. Okay, nucleus, what's going to happen? Step 1 is prophase. We're going to condense the DNA so we can move it around. We're going to get rid of that nuclear envelope so we can grapple onto it with microtubules. Metaphase, we're going to grapple onto it and move that condensed DNA around until it makes a line called the metaphasic plate. Okay? Anaphase, we're going to physically make those microtubules shorter and rip apart the DNA such that each side of this larger cell gets an even complement of DNA. Telophase, we're separating the DNA, now we're going to reform the nuclear envelope around it because we don't need to move it around, and we're going to decondense it so the DNA can interact better on a molecular level. Finally, that final separation is cytokinesis. Now, cytokinesis happens, or the cytoplasmic division happens a little bit differently in animal versus plant cells. In animal cells, we're going to form what's called the contractile ring. Okay? It's made of actin, and it's going to squeeze, get shorter, shorter, smaller, and smaller, and as it gets smaller and smaller, think of a belt tightening, right? And you get it smaller and smaller, and it's going to pinch the cell in the middle and pinch off the 2 such that we have 2 cells. And this is what we see in an animal cell. Mitosis is complete. This contractile ring of actin starts to get smaller and smaller and smaller and eventually pinches off to form 2 cells. Now, in plants, it's a little bit different. We're going to have a cell plate form. And this is a series of small pockets. And as these small pockets grow, they're going to form a contiguous plate. So plants do this cytokinesis with the formation of a cell plate. Okay. So here's our newly divided cell, and the blue are little clusters, little vesicles that are formed. Over time, those vesicles start to grow and start to fuse with nearby vesicles, making the cell plate. Eventually, that cell plate expands to an even larger structure, forms a partition, and then forms the 2 cells. Finally, we talk about cell cancer because with mitosis, uncontrolled mitosis is cancer. Okay? And how does cancer come about? When the checkpoints that control cell replication stop. Because we showed interphase, right? Well, a lot of times a cell will stop at a point before it gets into interphase, and that's it. That's it. It will not continue. But when a checkpoint, when a cellular control mechanism breaks down, you can enter into uncontrolled cellular replication. We talked about that in class. We talked about this in class, and that should be the review.